Hello everyone. Welcome to today's live broadcast, Learning Biology from Networks and Their Structures. I'm Julianne Chiat of LabRoots, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive. We encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. This webinar has been approved for continuing educational credits. Please click on the Continuing Education Credit button at the bottom left corner and follow the process to receive your credit. I would now like to introduce today's speaker, John Platig. John Platig received his PhD in physics from the University of Maryland. His thesis focused on the application of complex network methods to biological data sets, with an emphasis on understanding how errors in edge identification affect network properties. In conjunction with his physics training, he was a Cancer Research Training Fellow at the National Cancer Institute, working to identify potential therapeutic targets from a reconstructed gene regulatory network in diffuse large B-cell lymphoma. In 2013, John started as a postdoctoral fellow at the John Quackenbush at Dana-Farber Cancer Institute. He is currently working on the network inference and clustering methods to better understand genetic and other factors that drive phenotypes. I will now turn it over to John Platig for his presentation.
Thanks, Julianne. So today I'd like to talk about learning biology from networks and their structure. And what I hope to convince you of is that when we reconstruct or analyze networks in biology, that there is not necessarily a priori an important feature set, but rather that we have to take that network structure and think about it in the context of the biological data that we're using to make that network. And so the first half of the talk, I'd like to discuss understanding phenotypes using eQTL networks. And then the second half of the talk will be about elucidating drug response in tuberculosis with gene regulatory networks. So two very different sort of multi-omic network approaches. The one thing that we'll see throughout, though, is we'll use a method called CONDOR to try to model this biological regulation as a network system. So before I jump into the eQTL networks, briefly, CONDOR stands for Complex Network Description of Regulators. It's a method that we developed here in the Quackenbush lab, and it takes as its input a bipartite network, which is just to say that you have a network with two classes of nodes. They're represented here as red squares and blue circles. And a network is bipartite if you only ever have links between nodes of different classes. So the red squares only go to the blue circles. And our interpretation in sort of biological context of this bipartite network is that one class of nodes will be the regulators and the other class of nodes will be the targets. And in the first half of the talk in the eQTL networks, the SNPs will be our regulators and the gene expression of genes will be our targets. And in the second half of the talk, looking at the tuberculosis gene regulatory network, the regulators will be transcription factors, and again, the targets will be genes and their gene expression. So once you give CONDOR a bipartite network, what it does is then clusters that network using a community structure detection algorithm, and the method for using that that it uses under the hood is a bipartite modularity maximization. And sort of simply put, the goal is to identify groups of nodes that have more links within their community than that you would expect by chance, given the degree distribution of that network. And so you can sort of formally assign a quality function, Q, which is the modularity, and that's just the fraction of network links inside a community, and then you subtract that based on the fraction of links you would expect by chance. And so one of the sort of modifications from the more traditional modularity maximization is that we use a modularity shown in sort of the second reference here at the bottom, where the fraction of links expected by chance is altered to account for the bipartite nature of the network. So we never expect that two targets will share a link. So once you've done that, once you've clustered your network, you can then look at the nodes inside of those communities. And I should specify that each node goes into one and only one community, and every node is assigned to a community. And our intuition here for identifying what we call community cores is that not all nodes in the community are equal in terms of their contribution of the formation of that community or cluster. So, for example, the node that I've shown here with this green arrow is connected to all four of the blue nodes in its community in the center community. And so it seems to be very important for the formation of that community. Whereas the second node that I've highlighted with the second green arrow on the right here is only connected to two of the three nodes in its community. And so the intuition is that we'd like to say that this first node is actually much more important. It's sort of in the core of the community that it's a part of. And so we can formalize that by assigning it a core score, which is QIH. So it's the core score for node I in community H, and it's simply just the fraction of the modularity contributed by node I and then divided by the modularity of community H, which is the community that node I is in. So then what you can do is assign a score to all of the nodes in your network based on how central you think they are to the communities that they're in, and then use that to ask whether or not core nodes are more important in terms of functional enrichment or whatever it is that your biological network is trying to model. So I should also mention that CONDOR is available as an R package on GitHub, and feel free to email me if you have questions about that. There's a tutorial available as well. So our first application of CONDOR was to understanding genotype-phenotype relationships with eQTL networks. And the intuition for a sort of network approach to eQTLs is, I think, exemplified by the following question and the paper below it, which is how many SNPs affect complex traits? So this is a Nature Genetics paper from a couple of years ago, and it's a GWAS study looking at 253,000 individuals, and across this huge population size, they identified 697 variants that met genome-wide significance for being associated with height. And the thing that I think is particularly interesting is that they found that if they use the top 2,000, 3,700, or 9,500 SNPs, they were able to explain 21%, 24%, and 29% of the phenotypic variants. So what that means is that as we get larger and larger sample sizes and are able to explain more and more of the phenotypic variants, we actually need more and more SNPs to do this. And any one SNP is not going to have a huge explanatory power in terms of its phenotypic variants. So, there have been many calls recently for collecting, we need 10,000 samples to understand some complex disease, cancer, or what have you, or maybe 100,000 samples. But I think the question that we need to answer is, what do we do with 9,500 SNPs that explain a decent fraction of the phenotypic variants? But I can't go down the hall and ask someone to make a mouse model for all 9,500 SNPs. And I certainly can't have them start doing that in combination, where they pick SNP1 and SNP100 and make a mouse model for that. So I think what there's a need for is methods that essentially embrace this complexity. And this is another paper that came out recently that I think exemplifies this need as well. This is a GWAS study with 339,000 individuals, and they show that the top 97 loci account for 2.7% of the BMI variation. And so even the best hits only explain a fraction of the variation. So this suggests that you have many SNPs of weak effect that are working together. And so what we wanted to do was design a method that embraced this complexity at the outset. We knew that there would be lots of SNPs working together. So as we are a group that likes to use networks, our initial instinct was to use a network approach to look for groups of SNPs working together to try to influence cellular function. And so we did that by using sort of the most immediate phenotype in the cellular context, which is to link SNPs to gene expression via eQTL analysis, where eQTL analysis is just essentially a linear association between the SNP genotype and the gene expression of the gene you're interested in. So then what we did is treat eQTLs as a network of links between SNPs and genes, which then gives us a bipartite network. So as I mentioned before, the SNPs in this case are our regulators and the genes are our targets. And then the links that we draw between them are from the eQTL analysis. And those are the ones that are, I'll show in the next couple of slides, the ones that meet an FDR less than 10%. So there's a great data set actually for trying this method out, and that is the data released by the GTEx consortium. So this is data from their version 6, and we looked at eQTLs from 12 different tissues. There are 450 individuals represented. There are 4,900 samples, and we used the imputed genotypes and RNA-seq that they provided. We did some basic genotype filtering. We kept only SNPs that had a minor allele frequency greater than 5% across all of the tissues, since for each tissue has a different number of genotyped individuals and matching gene expression. So we made sure that we had a 5% cutoff for all of them. And then we used matrix eQTL, which is a very powerful R package for doing cis and trans eQTL mapping. So our thinking was we wanted to embrace all of the complexity of these associations. So we looked at cis and trans together. We then kept as links in our bipartite network those eQTL associations that had an FDR less than 10%. So then we get our bipartite network out. We can run CONDOR and cluster these things. And then we can ask, what sort of functional annotations do we find in these clusters? And I should mention that one of the advantages to embracing sort of the bipartite nature of these things is that we get clusters that have both SNPs and genes together in the same community. So you can interrogate the genes in the community, ask what is their enrichment, their role, their functional annotation, and then you can switch and do the same thing for the SNPs. So we ran on all 12 tissues, CONDOR, and did this bipartite modularity maximization. And so the modularity values that we got out, you can see here, and the modularity runs between 0 and 1, where 0 implies that there's no community structure, and 1 would imply essentially perfect community structure. And you can see that these networks are highly modular, with values between 0.75 and 0.95, which is fairly high if you look in the complex networks literature. And just to give you a sense of how modular these networks are, what you're looking at here on the right is the entire network for brain. Each dot in this figure represents a link, and the black links represent connections between SNPs and genes that are in different communities. And we've arranged this so that genes in the same community are next to each other and SNPs in the same community are next to each other. And then the colored links here represent SNPs and genes that are in the same community together. And so you can see that there's a strong block-like structure here. But then, of course, this is sort of a beautiful network, but does it have any interesting biology? So our first step for interrogating that is to ask whether or not any of the genes in these eQTL communities are functionally enriched. So for all of the tissues and all of the communities in each tissue, we performed a sort of basic Fisher's exact Go enrichment using the GoStats package in R. And this is a heat map of the results. So each column is a particular community in a given tissue. And each row then is a biological process from the gene ontology. And these have been clustered based on the minus log 10 of the p-value from the test that you got out. And so what we see are that even across different tissues, we find that communities in these different tissues cluster based on similar biological processes that make sense. For example, we have skeletal muscle and the tibial artery have communities that are enriched for muscle development and contraction. We have a number of different tissues, including skin, esophagus mucosa, that are enriched for epithelium development and keratinization. We find that cells that have sort of high turnover, fibroblasts and esophagus mucosa, are enriched, have communities that are enriched for mitosis. And then something that was interesting, although not surprising if you study the eQTL literature, is that we actually found an adaptive immune response community and enrichment for that in all 12 tissues that we studied. And then we also found a variety of Go terms related to the regulation of transcription in 8 of our different communities. And then sort of reassuringly, in one of our brain communities, we found enrichment for nervous system development and synaptic transmission. And I should mention at this point, too, that we did not feed any prior biological knowledge into this. We just did a complete cis and trans eQTL analysis. We clustered the results of that and then asked whether or not that clustering was overrepresented for biology. So we didn't use any sort of pathway annotations or anything to filter what we considered in our network. So it really is a data-forward sort of approach to modeling this. Now, one question you might ask is whether or not these communities simply just represent a particular location on a given chromosome. And so what we did then, this is, again, this brain network that I showed before. This is community 2 that's blown up. And what you're looking at here is a Circos plot for all of the links in that community. And the genes are highlighted. The red links that are connecting across in this Circos plot represent links that are connected to genes that contribute to this nervous system development and synaptic transmission. And what you can see as well is that this enrichment pulls genes from a variety of different chromosomes. So this functional signal is not coming from one specific location on the genome, but rather a multitude of different chromosomes, and that this eQTL analysis seems to be pulling that together into a community. So as I mentioned before, the other advantage of looking at this bipartite community structure approach is that we can then interrogate the SNPs. So as a first pass, our question was, where are the GWAS SNPs from the NHGRI GWAS catalog? So we looked at the degree distribution for all of the SNPs in our network, which are shown here in black. And you can see that they follow a very broad-tailed distribution, probably not exactly power law, but certainly power law-like. And then if you look, you can see that there's a slight overrepresentation in the middle of the plot for the GWAS SNPs, which are in blue. So you can see they tend to have a slightly higher frequency in this intermediate degree distribution. And so the degree in this context for a SNP is just the number of genes that it's associated with in the eQTL analysis. And that's a little more clear here, where on the y-axis we're plotting the observed degree for the GWAS SNPs over what you would expect given the degree distribution. And so what you can see here is that there seems to be a cutoff past about a degree of 10. And so what we're interested in then is, given this enrichment in this intermediate degree range, are these SNPs also likely to be cores, the GWAS SNPs? So we plotted here the core scores for all of our GWAS SNPs and compared those to all of the non-GWAS SNPs. And what you can see is that the median for the GWAS SNPs is certainly higher than for the non-GWAS SNPs. And the p-value here you're looking at is for the Wilcoxon test. So it looks like, at least in brain, that our core scores are higher for the GWAS SNPs than for the non-GWAS SNPs. And they seem to not be hubs, with one potential outlier. And so if we look across all 12 tissues, we see a very consistent signal, which is that you get this enrichment for intermediate degree, but you really don't see any global hubs in these for the GWAS SNPs, even though we observe them in the network. So given this sort of signal in the GWAS for core scores, and since we're using ostensibly healthy tissue from the GTEx consortium, what we wanted to do was, one, ask whether or not they have higher core scores, which we can see across different tissues here, It's not true for all 12 of our tissues, but it does seem to be true for many of them. So for example, fibroblasts, esophagus mucosa, lung, and breast, artery, tibule, and aorta. And some of that may have to do with the fact that the variation in the signal from tissue to tissue may have to do with the fact that we're using the NHGRI GWAS SNPs. But we're also interested in looking at the functional annotation for these core score SNPs. So what we did is we pulled all of the data from RegulomedDB, which is a resource that catalogs SNPs and then uses ENCODE data and the epigenomics roadmap data to try to categorize the putative functional impact of a given SNP. And so we recategorized those slightly so that they didn't depend on whether or not they were an eQTL, since all of our SNPs in these networks are already eQTLs. And so this top category A is the one that has the most confidence for a SNP being functional. So it has transcription factor binding. It has, from ChIP-seq data, it has a matched transcription factor motif for that binding site, a matched DNase footprint, and a DNase peak. And then as you go down, there is less and less data to support that a SNP is functionally relevant. And so I here is no information, which just means that there was nothing in RegulomedDB for that SNP. However, it was an eQTL in our network. So what we wanted to ask was whether or not core scores were more likely to be functionally annotated. And so here on the x-axis, you're looking at the odds ratio. And on the y-axis, you're looking at these various categories. And what you can see is that certainly in brain, there appears to be a consistently positive greater than 1 odds ratio for our core scores. And you can see that for some of the ones that are less confident, it's really a function of the number of SNPs in the category. So on the right-hand side, you're looking at the number of SNPs that were eQTLs in our network, and we're also in the category on the left in RegulomedDB. So this signal also seems to persist across these other tissues. So I'm showing you a number of examples. Brain is certainly one of the stronger results, fibroblasts as well. Lung and aorta also have strong positive signals for core scores being enriched for functional annotation in most tissues. And one caveat that I should list for this result is that RegulomedDB pulls these things from ENCODE, so there is going to be fibroblasts we probably have slightly more confidence in because we have lots of those tissues in ENCODE. And so there are, of course, limitations to using that database. So this was reassuring for treating eQTL networks as bipartite graphs, seeing that we saw the strong modular structure. But we wanted to apply this to a disease phenotype as well to try to get a sense of whether or not it could tell us something about disease biology. So we looked in COPD, which is chronic obstructive pulmonary disease. And COPD is the third leading cause of death in the US. It's commonly known by its subtypes, which are emphysema and chronic bronchitis. And from a genetic standpoint, one of the things that's interesting about studying COPD is that the environmental effect that leads to this disease is fairly well known, and that's cigarette smoke. So cigarette smoke, especially in the United States, is the leading cause of COPD. However, only 25% of smokers get COPD. So we thought that this eQTL network approach would be well suited to studying this. So we pulled data from the Lung Genomics Research Consortium, the LGRC, and that was a collection of lung tissue from 163 individuals, 126 cases with COPD, and you can see their distribution of disease severity in the rightmost column here, which is the gold stage, which is a clinical mark of disease severity. And then we compared those to our controls. And the other thing that was nice here is that we also had a smoking history for these individuals. So we did that. We took these 163 matched genotype and gene expression samples, and then did the matrix eQTL approach, looked at all cis and trans, pulled them together into a bipartite network, considered everything an FDR less than 10% to be links in our network and ran CONDOR, and then asked whether or not the things that we're observing in the GTEx eQTL networks were similar. So one thing that we found that was very similar with the GTEx tissues was that the SNP degree distribution seems to be fat-tailed. And in this case, it may actually be power law distributed. If you consider the top 10 connected components, then there's strong evidence that it is power law distributed. However, if you include all of the links, the probability that it's power law distributed is about 0.8. We did also observe that it was, again, highly modular. So you're looking at the sort of dot plot of the community structure on the bottom here, and the plot above that is just sort of a pretty visualization where you can see that there are many links within the communities and not very many that are going between different communities. And the modularity that we got was consistent with the values that we saw in eQTL networks in GTEx with a modularity value of 0.78. And we also found that if you look in the bottom left here, that NHGRI GWAS SNPs also seem to be enriched for this sort of intermediate degree range. So the red SNPs that you're looking at here are the GWAS SNPs, and then the black SNPs, the black dots, excuse me, are all SNPs in the network in their degree. But since we're looking at COPD, what we wanted to do then was ask whether or not there was a particular network feature for the GWAS SNPs from COPD. So what we did is, working with colleagues at Brigham and Women's Hospital who study COPD, they recently published a paper that's a meta-analysis that has approximately 14,000 different, approximately 14,000 individuals, and they did a GWAS for COPD. We took that and asked for all of the SNPs that were in our network. We computed a p-value based, or an FDR, excuse me, based on all of the GWAS SNPs in our network that had a reported p-value, and 30 of those had an FDR less than 5%. So then what we asked was whether or not these SNPs had any sort of particular features in the network. And the first thing we did was look at the core scores and asked whether or not they were higher for the COPD GWAS SNPs compared to the non-GWAS SNPs in our network. And we found that the median core score for these 30 SNPs was actually 20 times higher than the median for the non-significant SNPs. And another thing that we wanted to do here was to test whether or not this difference was a function of linkage disequilibrium. So you could imagine that maybe all of these SNPs are in strong LD, and somehow that's driving the connectivity of this network and driving the core scores that we're observing. So what we did is, for our original comparison here with FDR significant SNPs versus non, we replaced our GWAS SNPs with a matched set of 30 SNPs that had the same LD structure as our GWAS SNPs, and we randomly sampled sets of 30 1,000 times and asked whether or not this difference that we observed was a result of just the LD structure. And so what you're looking at on the right here is a histogram. We used the one-sided Wilcoxon test to look at this, and the red dot in the bottom right here is actually the true difference between our GWAS SNPs and their core scores and the non-GWAS SNPs. And then the histogram here is just our permuted values. And something I didn't show, but that we also tested, is whether or not this is a function of distance to the nearest gene. And we found that that was not the case for that as well. Again, with a p-value less than 0.001, given 1,000 random samplings. So it seems like this core score approach does a good job of identifying sort of GWAS SNPs. What we found also is that the function for these 30 FDR significant GWAS SNPs in COPD were all annotated, with the exception for having putative function. And this is, again, coming from Regulum DB. So the results that we saw in the GTEx eQTL network seem to be fairly concordant with the same kinds of network signal that we're seeing in our COPD data set. So just as a recap, we see that these eQTL networks really do show strong community structure across tissues and phenotypes. These GWAS SNPs from the NHGRI GWAS catalog appear to be depleted from global hubs. However, the SNPs in the cores of their communities are more likely to be functionally annotated and that they are also more likely to be disease associated. So switching gears here, I'd like to talk about a very different kind of network, which is a gene regulatory network. And this is a project to try to understand PknA inhibition in tuberculosis. And so this was done in conjunction with 2 groups at Children's Hospital across the street from us. And the reason for looking at PknA is that PknA and PknB are essential kinases in tuberculosis. And they've been shown to be involved in cell shape control and cell wall synthesis. But the pathways regulated by these kinases are not fully known. So what we'd like to do is actually inhibit PknA and B. There's a drug for doing that, a small molecule, and then try to elucidate what the downstream effects are using a gene regulatory network approach. So what we have then are a series of experiments in cell lines. So this is the H37RV tuberculosis cell line. And we have 2 compounds with what we call T4, which is the active version of the PknA inhibitor, AB inhibitor, and then a chemically inactivated version of that same inhibitor, which we call T10, which is our control compound. So then what we do is perform a time course and then collect RNA-seq at 12, 24, and 48 hours. And we have 9 samples for each treatment time point combination. And so what we'd like to do then is actually integrate that with publicly available ChIP-seq data for 143 transcription factors that have been chipped in tuberculosis, and also protein-protein interaction data that is also available on the string database. And so our method for integrating all of this is a gene reconstruction algorithm called PANDA, which was developed by Kimberly Glass, who's a former postdoc in our lab. And if you're interested in more details and how to use PANDA and really sort of the nitty-gritty of how it works, I would recommend going back and looking at the talk this afternoon from my office mate, Marika Kuyer, who discusses how PANDA works in some detail and also provides a number of examples for how you can use it in human diseases as well, cancer, for example. But for our purposes here, I'll just briefly recap how PANDA works, which is that it takes interaction data. So in this case, we're interested in transcription factor protein-protein interactions, and then ChIP-seq data from 143 transcription factors in tuberculosis, and then our expression data, which was collected from our time course that I described over here on the left. And so it produces 3 sort of initial networks. The ChIP-seq data is a sort of initial seed as our regulatory prior, if you will. And then the expression data is turned into a co-regulatory network for the genes, which is just a Pearson correlation matrix as our initial guess for that. And then the transcription factors are the initial guesses for their interactions from this PPI network. And so then what PANDA does is it uses a message passing approach to ask what is the most concordant regulatory network that gets positive inputs from all 3 data types and tries to get a sort of best representation that is most concordant across all 3 data types. And what PANDA produces is a weighted bipartite graph. So what we can do then is compare the node degree, for example, from this output, where again the degree is just the number of edges that are coming out from a given node, and these are weighted. So we can look at the transcription factors and ask whether or not the sum of their outgoing weights is different. And then we can also use CONDOR to detect communities and calculate core scores and ask whether or not there are differences between these 2 things. So our first step is to look at differences in transcription factor out degree. And again, the out degree is just the sum of the outgoing edge weights for each transcription factor in each condition. So you sum over all the weights for a given transcription factor in each condition and ask how different those 2 things are. And what you're looking at on the right here is the distribution of differences for our 143 transcription factors. And you can see there is a sort of a slight skew for a negative difference. Again, reminding you that T4 is our active compound and T10 is our control. So there seems to be slightly higher out degree for the transcription factors in the control compared to the treatment. And one way to think of that is that these sort of negative values mean that the expression of the genes in the regulon of the transcription factor are less correlated. So they're sort of decohering in response to this treatment of PknA inhibition. So then we wanted to see what were the most significant changes in out degree. So you're looking at the transcription factors here, ranked by the magnitude of change in their out degree. And then the p-values here are permutation p-values where we've shuffled the gene labels on the gene expression and rerun PANDA completely. So we did this 1,000 times. We shuffled the genes, and then recalculated a new regulatory network based on those shuffled gene labels. And so this is the empirical p-value for those. They're all less than 0.1. In fact, all of them are less than 0.05, with the exception of this 10th one right here at the bottom. And then the rightmost 3 columns are the log-fold change in the differential expression from the RNA-seq, and the asterisk here means that they had an FDR less than 0.05. And so you can see there's some of the genes are these transcription factors are differentially expressed, but not all of them are. And so then we went and asked whether or not these top differentially targeting transcription factors are actually interesting in the context of PknA inhibition. And we actually found that the 4 that have gene names here are actually all associated with signal transduction, cell wall function, or lipid metabolism. And again, PK and B have been shown to affect cell wall morphology and cell wall function. And then RV0081, our most differentially out degree transcription factor, has actually been shown to be a master regulator in a gene regulatory network reconstructed from TV looking at hypoxia. And so KSTR as well is involved in fatty acid metabolism and cholesterol, so it seems to be cell wall associated. LSR2 has been previously shown to be associated with cell wall function, and CSOR is thought to mediate oxidative stress response. So all of these seem to make sense in the context of disruption of the cell wall. So one of the other things that PknA and B do is they affect the phosphorylation of other kinases. So as a validation of our top differentially out degree transcription factors, we looked at whether or not any of these transcription factors were differentially phosphorylated in response to PknA inhibition. And so what you're looking at here is the changes in out degree for all 143 transcription factors, and then those that are labeled in red are differentially expressed phosphopeptides. So they're differentially phosphorylated with a log-fold change greater than 0.5 and an FDR less than 0.05. And what you can see is that our most positive change in out degree, so the transcription factor whose out degree increased the most, is actually one that is differentially phosphorylated. And the log-fold change for this CSOR is actually negative 1.8, which is one of the top differentially phosphorylated transcription factors that we found. So we were also interested then in whether or not the whole class of differentially phosphorylated transcription factors were more likely to have a larger change in their out degree than the rest of the transcription factors. And so we did a permutation test to compare the ratio of medians, so the median out degree, change in out degree, excuse me, for our phosphorylated transcription factors, divided by the median out degree for our transcription factors that were not differentially phosphorylated. And we saw that the ratio of these medians is 2.5, and then we did a permutation test and found that that p-value is less than 0.024. So it seems that differentially phosphorylated transcription factors also have differential out degree, which we thought was a nice confirmation of our prediction from the network. So given this, we wanted to look at sort of the next level of organization up and compare the community structure. So this is, again, one of the dot plots. So each dot here represents a link between a transcription factor and a gene. And the colored links here represent transcription factors and genes that are in the same community together. And this is for the T10 network. And you can see that the modularity is not as high in this gene regulatory network than it was for any of the eQTL networks. So it only has a modularity of 0.5. And it's very similar in the T4 network as well. So the modularity there is also 0.495. So they're almost the same. One of the questions that we had then is that given that these two different networks appear to have very similar community structures, and if you just kind of look at these two figures for a little while, you can see that there doesn't appear to be very much changing between the two. And we were wondering then whether or not it was community cores that were driving this similarity. So what we wanted to ask was if something is a community core based on our core score in the T10 network, is it more or less likely to sort of change its membership in the T4 network? And so when we say here PknA be perturbed, that means that the transcription factor changes its community. So it's no longer if it's in a T4 community that doesn't have the highest overlap with its T10 community, we say it's perturbed. And so what you can see here for the two distribution of core scores is that those that have much higher core scores are more likely to be unperturbed, and those that have lower core scores are more likely to be perturbed. So it seems that the cores are not being affected by this PknA and B inhibition. We then looked at this for the genes as well, using the core scores from T10, and we found that the result was the same, which is that things that are unperturbed are more likely to have high core scores than things that are perturbed. So it seems that this gene regulatory network is really trying to maintain its community structure in response to this PknA/B inhibition. We then tried to look at drug-specific community enrichment. So we looked at the genes in each of the communities in our T4 and our T10 networks, and we found that there's some similarity between them. So for example, oxidative phosphorylation shows up both in T4 and in T10. But one of the new functions that appeared in T4 that was not in T10 is an enrichment for response to stress. And in fact, one of the differentially phosphorylated transcription factors that we identified was also involved was located in this community, and it's a heat shock protein, so you think that it's involved in response to stress. So we did find some signal in the community approach, but it wasn't nearly as fruitful as looking at the differential out degree for the transcription factors. And so as a recap, for the TB section, we found that differentially phosphorylated transcription factors, especially CSOR, are more likely to be differentially targeting, so have a change in their out degree in our network model. And we think this may reflect the effect of PknA/B on phosphorylation of other proteins. And we found that gene regulatory networks don't appear to be as highly modular as the eQTL networks. However, the degree of nodes in the gene regulatory network seem to be much more useful than the global degree in the eQTL network. And then finally, we found that the community cores, both for the transcription factors and the genes, appear to be robust to PknA/B inhibition. And so with that, I'd like to thank my collaborators, my PI, John Quackenbush, Maud Finney, and Joe Paulson, who worked with me on the GTEx eQTLs, Albert Young, who was a postbac in our lab, who did some great work on the TB regulatory network, and then our collaborators at Children's Hospital on the TB project, Bob Husson and Hanno Steen, Xavier Corrette, Michaela Hemmel, and all of our collaborators at Brigham and Women's who worked with us on the COPD eQTLs. And so with that, I'd be happy to take any questions. Thanks.
Great. Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them into the Q&A box, which can be found by clicking the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, In the context of thermodynamic cycles of cell type differentiation and phenotypes, what is the difference between an eQTL network and a network that is mediated by microRNA flanking sequences?
I think the difference here is that I think the difference here at the biological level is just that we're looking at sort of polymorphisms that are disrupting binding or regulation in some way. I think at a higher level, the network is sort of flexible in the sense that if you can understand this as a bipartite network and model it as such, then I think there is some similarity there. Although I think that, you know, for the data that we've presented here, we can't really speak to the thermodynamics of the cell type differentiation. That would be an interesting problem to study, but I think that we would need much sort of finer grain experiments and better cellular assays to assess that.
If you co-authored a paper that linked RNA expression data to the protein state of cells, please comment on any links from energy-dependent cycles of protein biosynthesis and degradation to healthy longevity via the innate immune system, the physiology of reproduction, and supercoiled DNA. Alternatively, do you know any possible links from virus-driven energy theft to pathology?
So I can't speak to any sort of virus-driven energy theft. What we looked at here in tuberculosis, in the bacterial sense, we do have protein expression for the experimental setup that I described earlier with this PknA inhibition. One thing that we found that I thought was interesting is that the RNA expression was not very well correlated with the protein. If you correlate a gene's RNA level and its protein level at the same time point, the Pearson correlation is about 0.2. However, that correlation doubles to about 0.4 if you look at a time lag. So if you correlate the mRNA at 12 hours, for example, and the protein expression at 24 hours, you see much better correlations. So that's, I think, as much as I could speak to on that question.
In the tissue-specific eQTLs that group together, are there known genetic interactions for these eQTLs?
Yeah, that's a great question. There aren't any that I know of, although to be honest, we have not yet tried to look for them. I think epistatic interactions in particular would be a really fruitful way to go. From the network modeling perspective of that, it would violate sort of our bipartite structure, but there are lots of, I think that would be an example of where you would want to change your network model to reflect what you think is going on biologically, because I think that a lot of what's going on in these eQTLs is probably a function of epistatic interactions, and that might help explain some of this phenotypic variance in and the absence of it in these GWAS studies where you have 330,000 individuals.
There are no more questions. I would like to once again thank Dr. Platig for his presentation. Do you have any final comments?
Very much for attending.
Thank you very much for attending.
Thank you for attending.
Today's webcast will be available for on-demand viewing through May 12, 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.